Welcome back, everyone, for another deep dive. This time we're going to be looking at something that you probably haven't thought about too much.
Yeah.
It's habanero peppers.
Habanero peppers.
But not the peppers themselves, the fruit part that we eat.
Yeah.
We're looking at the stems and the leaves.
The parts we throw away.
Exactly. So the stuff that most people just chuck in the compost.
Yeah, into the garbage or whatever.
Yeah, exactly. And this is from a research article that I found super interesting, and I know you did too.
I did. It's so cool.
And it is called Antioxidant Capacity, Vitamin C, and Polyphenol Profile Evaluation of a Capsicum chinense byproduct extract obtained by ultrasound using Eutectic solvent.
That's a mouthful.
It is a mouthful. So Capsicum chinense, that's the species that we know as the habanero pepper.
Yeah.
But what was so interesting to me about this is they are using these stems and leaves and extracting all these good compounds.
Like we would from the pepper itself.
Yeah.
So we're finding all these wonderful things that are kind of being wasted.
Exactly.
When we just toss those stems and leaves.
Yeah, and the varieties that they used were Mayapan and Jaguar.
Oh, cool.
So I don't know if you've heard of those.
Oh, I haven't, but I am intrigued.
But they're extracting mostly polyphenols and vitamin C.
That's great. I mean, those are both
very
valuable compounds that we need.
Yeah, and the way that they're doing it is
interesting.
just pretty cool, too. They're using something called NADES.
I love it.
Which stands for Natural Deep Eutectic Solvents. And it's a green extraction technology.
Which is great because a lot of the times when you are extracting chemicals and compounds like this from plants, they use pretty harsh chemicals.
Yeah.
That aren't so good for us or the environment.
Right. And so in this case, the NADs are things like choline, chloride, and glucose.
Okay.
And sometimes even honey.
Honey. That's really cool. I don't know how they even figure this stuff out, but it's amazing.
Right. It's incredible.
What a great idea.
And so they're comparing this to the results that you would get from using methanol.
Okay.
Which is what's more commonly used.
Right. More of a standard method.
Yeah, exactly.
So they're comparing these two to see how they do.
Yeah. And what were some of the things that they found?
One of the most interesting things was that they found way higher concentrations of polyphenols in the NADS extracts than the methanol.
Wow.
Which is really interesting.
So it's not just that it's better for the environment.
Right. It's not just about being green and sustainable. It's about getting more of those good compounds.
Exactly. Yeah. Which is so cool. And specifically, they were talking about catechin.
Oh, wow. Yeah.
Which was almost non-existent in the methanol extracts.
And catechin is a powerful antioxidant.
Yeah.
It's been linked to all sorts of health benefits.
Yeah. So getting more of it is a good thing. It's kind of crazy that it's almost non-existent in the methanol and that you can get so much of it in these NA.
Right. And it's really neat because it's this ability for NAs to form hydrogen bonds with the polyphenols that makes them such a great way to extract them.
Okay.
So they're able to kind of pull them out more effectively than the methanol can.
So what are some of the other things that they found in there?
Well, they found all sorts of great polyphenols.
Okay.
They found gallic acid, which is known to have some pretty potent anti-cancer properties.
Wow.
They also found protocatechuic acid.
Okay.
Which is another great antioxidant.
Yeah.
That's been linked to reduced risk of heart disease.
Wow.
And then they found things like caffeic acid and ferulic acid.
Okay.
Which are also great antioxidants.
So he said?
And they're all found in different concentrations.
Yeah.
Depending on the variety of pepper and the extraction method.
It's like a whole pharmacy.
It is.
Yeah. In these stems and leaves.
It really is. It's amazing what nature can provide.
And so when they're talking about antioxidant capacity, like how are they measuring that?
Well, they used a couple of different tests.
Okay.
One is called the ABTS assay.
Okay.
And the other one is called the DPPH assay.
And those are both?
And these are standard tests that are used to measure the antioxidant capacity of different substances.
So what are they actually measuring?
So essentially they're measuring the ability of the antioxidants in the extract to neutralize free radicals.
Okay.
So free radicals are these unstable molecules that can damage our cells and contribute to aging and disease.
Yeah.
Antioxidants can donate an electron to these free radicals, which stabilizes them and prevents them from causing damage.
So it's kind of like...
So the ABTS and the DPPH assays are basically measuring how effectively
Yeah.
the antioxidants in the extracts can do that.
Like a boxing match?
Yeah, kind of like a boxing match or a test of strength to see how well those antioxidants can fight off those free radicals.
And so the results that they got
Yeah.
for those were also pretty interesting.
Yes. So they found that both the NADES and the methanol extracts
Oh, okay.
had high antioxidant capacity.
Okay.
But they differed in the type of antioxidant activity.
What does that mean?
So the NADES extracts were more effective in the ABTS assay,
Whoa. Okay.
whereas the methanol extracts were more effective in the DPPH assay.
Oh. So it's like,
So it suggests that the types of polyphenols present
yeah.
in each extract were different,
Yeah.
and they were interacting with those different tests in different ways.
So they're better at fighting different things.
Exactly. They each have their own strengths and weaknesses.
Okay. So maybe that's a good place to pause for now.
Sounds good.
And we can pick it back up in the next part.
Yeah, I'm excited to keep going.
Me too.
This is fascinating stuff.
Okay. See you next time.
See you then. Back again.
Ready for more on these habanero pepper byproducts?
Oh, yeah. I'm totally hooked on this.
It's so cool.
I mean, who knew?
Yeah.
All this good stuff was just going in the trash
Right.
this whole time.
And it's like
a
filled.
secret world.
The NADES, I am so fascinated by this, honey.
Well, yeah.
Come on.
It doesn't...
Who figures that out?
I know. That's what...
Brilliant.
It's brilliant.
Yeah.
So last time we were talking about
Yeah.
how the antioxidant capacity is measured
Right.
using the ABTS and the DPPH assays.
Yeah.
And
they were finding different results.
yeah, they were finding
Given the method.
that the NMAs were better in the ABTS
Right.
and the methanol was better in the DPPH.
Exactly.
So what exactly are those
Oh,
measuring?
it's really complicated.
Yeah.
But they're actually pretty straightforward.
Okay.
So basically what they're doing is they're measuring the ability of the antioxidants in the extracts to neutralize free radicals.
Okay.
So think of free radicals as these little troublemakers.
That are running around in our bodies,
Okay.
causing damage to ourselves.
Yeah.
They're unstable molecules
Right.
that are missing an electron,
Okay.
and they're constantly trying to steal electrons from other molecules.
So they're just like causing chaos?
Exactly. They're causing
Yeah.
chaos, and that damage can lead to all sorts of problems like
Right.
aging and disease.
Yeah.
So antioxidants come in,
Okay.
and they're like the superheroes.
Okay.
They can neutralize those free radicals
Yeah.
by donating an electron to them.
Oh, so they're like...
So basically, they're stabilizing those free radicals,
Yeah.
preventing them from causing damage.
Taking one for the team.
Exactly. They're sacrificing themselves to protect ourselves.
Okay. That makes sense.
So the ABTS and DPPH assays are basically measuring how effectively the antioxidants in the extract can do that.
Okay.
How well can they neutralize those free radicals?
Yeah.
So it's like...
And it's interesting because they use
Yeah.
different free radical molecules
Okay.
in each assay. So they're testing it against different...
Exactly.
Yeah.
They're testing them against different opponents
Okay.
to see how well they perform.
And that's why they're better.
And that's why we see...
Like different ones.
Yeah, that's why we see those differences
Okay.
in the results.
And so...
Depending on the extraction method,
Yeah.
you're going to have different types of antioxidants present.
Okay.
And those different types of antioxidants
Yeah.
are going to perform differently in those different assays.
Oh, that's so cool.
Yeah, it's really fascinating.
So this is making me think about like
Yeah.
all those supplements that you see like
Right.
in the store.
Like antioxidant this
Yeah.
and antioxidant that.
Yeah, they're like, oh, this is full of antioxidants.
Exactly.
But it seems like it really matters.
It does what type
It matters what type
and how it's even measured.
and how it's measured. Exactly. It's not just about the quantity.
Right.
It's about the quality.
That makes sense. Of those antioxidants.
Okay.
So yeah, it's something to keep in mind.
So we have to be more discerning consumers.
Exactly.
Yeah.
You can't just take those claims at face value.
Wow.
We got to dig a little deeper.
So they were looking at the
Yeah.
polyphenols
Yeah.
and the vitamin C.
Right.
What about other things like carotenoids?
Well, interestingly enough,
Yeah.
they didn't find a lot of carotenoids
Oh, really?
in these extracts.
Huh.
And that's probably because
Yeah.
NADES are really good at extracting hydrophilic compounds.
Okay.
Things that like water.
Right.
Carotenoids are hydrophobic.
Right.
They don't like water.
So the
Well, the
NADES...
NADES are just not going to pull those out.
They're not going to pull those out as effectively.
Okay,
Yeah.
so it's not that the habanero stems and leaves don't have carotenoids.
Right.
It's just that the NADs
Yeah.
that they were using in this particular study
Right.
weren't the best.
Weren't the right tool for the job.
Exactly. They weren't the right tool for the job.
But there might be other...
Now, there might be other types of NADES
Right.
that could be used
Yeah.
to extract those carotenoids.
Cool. So it's just a matter of...
Yeah, it's a matter of finding the right NADES for the job.
Okay.
So it's really interesting because this just shows us that NAs are a very versatile tool.
Okay.
And we can kind of tailor them to extract different types of compounds.
That's amazing.
It is. It's really cool.
Yeah, it's like a chemistry toolbox.
It is. It's like a chemistry toolbox.
Wow.
And we're just starting to figure out
Yeah.
all the different ways that we can use it.
So they also found vitamin C.
Yes, they did.
In these extracts.
And again, the NADES extracts had significantly higher concentration of vitamin C
Wow.
compared to the methanol extracts.
So another win for NADES.
Another win for NADES.
And especially the honey-based NADES.
Especially the honey-based NADES. Right.
So why is the honey so good at pulling out the vitamin C?
Well, it has to do with the fact that vitamin C is a hydrophilic compound.
Okay.
Meaning it likes water.
Right.
And honey-based NADES are also very hydrophilic.
They're both water lovers.
Exactly. So they're able to kind of pull that vitamin C out more effectively.
Oh, that's so cool.
Yeah, it's really neat.
So we've learned so much already about these habanero pepper byproducts.
I know. I'm like blown away by
it's like
the potential here.
Whole new world.
It is.
So maybe that's a good place to pause again.
Okay.
Catch your breath.
Yeah, that's my breath.
And then we'll come back for the final part.
Sounds great. I'm excited to talk about what this all means.
Yeah. What are the implications?
Yeah.
All right. See you next time.
See you then.
Okay. So we're back for the final part of our habanero deep dive.
I'm ready.
Me too. And I have to say, like, I'm still kind of blown away by all this.
Me too.
It's like just the fact that all these like amazing compounds are in these parts of the plant that we just
throw away.
Throw away, yeah.
It's crazy.
And that honey.
Honey. Don't forget the honey.
I know. The honey.
So cool.
Is this like magic extraction tool?
It's pretty amazing.
It is.
Yeah.
So we've talked a lot about what they found and how they extracted it.
Right.
But I guess the big question now is like...
The so what?
Yeah, the so what? Like,
Right.
what are the implications of this?
Well, I think the implications are huge.
Okay.
I mean, this could really change the way we think about
Yeah.
food waste
Okay.
and resource utilization.
Okay.
Imagine a world where food producers no longer view these byproducts as waste,
Right.
but as valuable ingredients
Yeah.
packed with nutrients and bioactive compounds.
Like, we don't have to feel bad about chucking those stems and leaves in the compost anymore?
Exactly. Instead of composting them,
Yeah.
we could be extracting all these valuable compounds.
Right. And using them in other products.
Exactly. I mean, think about all the different food products that we could create
Yeah.
that are enriched with these extracts.
Oh.
We could have polyphenol-boosted smoothies,
Okay.
antioxidant-packed snacks,
Yeah.
baked goods. I mean, the possibilities are endless.
Oh.
And it's not just limited to food.
Okay.
We could also use these extracts in dietary supplements.
Oh, yeah. That makes sense.
Right.
Yeah, like a vitamin C supplement
Exactly.
that's made from habanero stems.
Instead of synthetic sources.
Wow.
So much better
That's amazing.
for you and for the environment.
Yeah, that totally aligns with like the whole trend of like the people wanting more natural products.
People want natural.
Yeah.
They want sustainable.
And sustainable, yeah.
This ticks all those boxes.
Wow.
And, you know, it's not just about the consumer products. I think this could also have a big impact on agriculture.
Okay. In what way?
Well, imagine farmers being incentivized to utilize the entire habanero plant, not just the fruit.
So they can sell those stems and leaves to like extractors.
Yeah. It creates a new revenue stream for them.
Yeah.
And it promotes more sustainable farming practices.
So it's like a win-win.
It is. It's a win-win.
For everybody.
For everybody.
But I imagine there are some challenges.
Oh, of course.
Too, right.
There's always challenges.
Like, how do we actually, like, implement this?
Well, one of the big challenges is scaling up the extraction process.
Okay.
Right now, NADES extraction is mostly done on a lab scale.
Okay.
To make it commercially viable.
Right.
We need to figure out how to do it.
Yeah.
On a much larger scale.
So it's got to move from the lab.
Exactly. From the lab to the factory.
Right.
And that's going to require some engineering, some innovation.
Right.
But I think it's definitely possible.
Yeah.
It's just a matter of putting our minds to it.
And then I guess another challenge is like consumers.
Right.
Like, are people actually going to like buy products?
Yeah. Are people going to be grossed out?
That are made from?
By the fact that it's made from?
Like what used to be considered waste?
Stems and leaves?
Yeah.
Yeah, it's a valid concern.
Yeah.
I think it's all about education and transparency.
So we got to teach people about this.
We got to teach people about the science behind it, the benefits.
Like...
We got to be transparent about where these ingredients are coming from.
Right. And that it's not gross.
It's not gross. It's actually
good.
Really good for you.
Yeah, and good for the planet.
Exactly.
Yeah.
So it's about changing that perception from waste to valuable resource.
I love that.
Right.
Well, I'm convinced.
Me too. I'm totally on board.
This has been
a fascinating deep dive.
Yeah, eye-opening.
Eye-opening for sure.
I'm so glad we did this.
Me too. It's amazing what you can learn when you start digging into
when you take a deep dive.
The scientific literature.
All right, well, thanks for joining me on this.
It's been a pleasure.
Habanero adventure.
Yes, a habanero adventure indeed.
And to all our listeners out there.
Yeah. Keep those compost bins handy.
You never know.
You never know what treasures.
What's in there?
Might be lurking.
All right, we'll see you next time.
See you later.